On a red desert 225 million kilometers away, a $2.7 billion machine is staring at a mudstone rock. Not just any rock. On its surface, there are tiny spots, black, white, arranged in strange patterns. Scientists call them leopard spots. In July 2024, the Perseverance rover, or Percy, as NASA engineers affectionately call it, discovered these spots in a rock named Chiaiva Falls. These aren't random markings. They have clear structure, a light-colored inner core, similar to the surrounding rock, but a dark outer rim, enriched in iron and phosphorus. And if scientists are right, this could be the clearest evidence of life beyond Earth that humanity has ever found. Because on Earth, structures similar to these are almost exclusively created by one thing: bacteria. But before we understand why these spots matter so much, there's a bigger question that needs answering. Out of 60 candidate sites on Mars, why did NASA choose this exact location to land? Why did they bet billions of dollars on an ancient impact crater called Jezero? To answer that question, we need to understand 4 critical things. First, Jezero Crater isn't just an ordinary impact crater. This is a paleo lake. An ancient lake that existed 3.6 billion years ago when Mars was warm and wet. Second, perchlorate, a chemical compound that on Earth some bacteria can use as an energy source to survive. But it can also be a killer, destroying organic molecules under ultraviolet light. So is it friend or foe? Third, Vivianite, a blue-green mineral formed when bacteria breathe iron instead of oxygen. And the leopard spots Percy found might be the fingerprint of this very process. And fourth, how do we know this isn't a false alarm like 1996? When NASA announced they'd found life on Mars, only to admit 26 years later they were wrong. This is the story of the science behind humanity's greatest hunt for life. And it all starts with choosing the right place to look. 60 sites, years of fierce debate, billions of dollars on the line, one ancient crater that could rewrite everything we know about our place in the universe. This is Jezero, and today you'll understand exactly why NASA chose it. From 2014 to 2018, NASA assembled the world's top scientists to do one thing: choose the landing site for Perseverance. 60 candidate locations, hundreds of meetings, thousands of hours of debate. The criteria were crystal clear. First, safety. No steep cliffs. No giant boulders that could destroy the rover during landing. Second, science. There must be clear evidence of past water. Third, diversity. Multiple rock types for sampling. And fourth, age. Old enough, at least 3.5 billion years, when Mars could still support life. Jezero passed them all. 3.6 billion years ago, Jezero wasn't the dry crater we see today. This was a lake, 500 meters deep, 45 kilometers wide. Water flowed in from the west through 2 ancient river channels, carrying sediments, forming a massive delta, exactly like the Mississippi River emptying into the Gulf of Mexico. On Earth, river deltas are the best places for fossil preservation. Why? Because sediments bury organisms very quickly, protecting them from decomposition, turning them into fossils over millions of years. If life ever existed on Mars, Jezero's delta is the ideal place to search for traces of it. But there was a hidden reason few people know about. A reason that connects directly to those leopard spots Percy would discover years later. That reason has a name: perchlorate. Perchlorate. A word most people have never heard. But for Mars scientists, this is one of the most important and most controversial compounds. In 2008, NASA's Phoenix lander detected perchlorate in Martian soil for the first time. And since then, we found it everywhere on the red planet. from the poles to the equator, from the surface deep into the ground. But here's the paradox. Perchlorate could be life's best friend, or it could be its deadliest enemy. On Earth, there's a special group of bacteria called perchlorate-reducing bacteria. These organisms don't need oxygen to survive. Instead, they breathe perchlorate. They take the perchlorate molecule, ClO4-, and break it down step by step, from ClO4- to ClO3-, then ClO2-, and finally ClO- plus oxygen. This process releases energy that bacteria use to live, to grow, to reproduce. Just like we breathe oxygen to power our bodies, these bacteria breathe perchlorate. And here's what's fascinating. Paleolake basins, ancient lakes like Jezero, tend to accumulate more perchlorate than other regions on Mars. Dr. Kenda Lynch, an astrobiologist at the Planetary Science Institute, has studied similar environments on Earth. Dry salt lakes in Chile's Atacama Desert, and she found bacteria surviving on perchlorate even in the harshest conditions. So, if Jezero had perchlorate 3.6 billion years ago, and if there were bacteria on Mars, this would be the perfect food source for them. But that's only half the story. Now, let's talk about perchlorate's dark side. When combined with ultraviolet radiation from the sun, which Mars has no magnetic field to block, perchlorate becomes an extremely powerful toxin. In 2017, a study published in Scientific Reports demonstrated this terrifying effect. Scientists placed Bacillus subtilis bacteria, a very resilient Earth bacterium, in a simulated Martian environment. They added perchlorate, iron oxides, and hydrogen peroxide, all readily available on Mars's surface. Then they exposed it to UV light. The result? Bacterial death rates increased by a factor of 10.8, not 10.8%, but 10.8 times higher than UV alone. Perchlorate under UV creates extremely reactive free radicals, molecules that can tear apart cell membranes, destroy DNA, and turn any organic molecule into dust. Mars's surface is a giant UV bath. No ozone layer for protection, no magnetic field for shielding. Perchlorate here is like a chemical bomb waiting to explode every time the sun rises. So how do we resolve this paradox? The answer lies in depth. On the surface, where UV, perchlorate, iron oxides, and H2O2 combine, that's a dead zone. But just a few centimeters below, the story is completely different. Down there, there's no UV. Perchlorate is no longer toxic. Instead, it returns to its role as a potential energy source. Scientists call this subsurface habitability. And this is exactly why rocks like Jezero Falls matter so much, because they were once buried deep in delta sediments, protected from UV, where perchlorate could feed bacteria instead of killing them. But perchlorate isn't the only compound bacteria can use. On Earth, there's another group of bacteria, specialists in breathing something completely different: iron. Let's return to those leopard spots Percy discovered in the Jezero Falls rock. These spots aren't random. They have very clear structure. The inner core is light-colored, similar to the surrounding rock, but the outer rim is dark, especially enriched in two elements, iron and phosphorus. And the ratio between them is nearly perfect. 1 part iron, 1 part phosphorus. This is exactly the chemical signature of Vivianite, a blue-green mineral with the formula F3PO428H2O. But what makes Vivianite special isn't its chemical composition, it's how it forms. On Earth, Vivianite is almost always formed through a process called iron reduction. And this process, in most cases, is performed by bacteria. Imagine how it works. A bacterium, for example Geobacter sulfur reducens or Shewanella lohica, attaches to an iron oxide particle in the sediment. This iron oxide contains Fe-cubed Ms iron in the +3 oxidation state. The bacterium doesn't eat iron in the literal sense. Instead, it uses iron as an electron acceptor. Through the cell membrane, using special proteins called cytochromes, the bacterium transfers electrons from inside the cell outward, donating them to the iron atom. When Fe-cubed Mi receives an electron, it transforms into Fe² Mi, iron in the +2 oxidation state. And this process releases energy that the bacterium uses to live, a process called chemosynthesis. No sunlight needed, no oxygen needed, just iron, water, and a bit of organic carbon. And when Fe² Mi is released into the water, it combines with phosphate and water to form Vivianite. Tiny blue-green crystals begin growing around the bacterial cells, like chemical fingerprints of life. What's wonderful about Vivianite is that it's extremely stable. Once formed, it can persist for billions of years without being destroyed. It's like a geological time capsule, preserving evidence of microbial activity from ancient times. But Vivianite can't form just anywhere. It needs very specific conditions. First, neutral pH, around 7. Too acidic or too alkaline, Vivianite won't be stable. Second, the Fe-P ratio must be balanced, close to 1:1. Third, the environment must be anoxic, no free oxygen, because oxygen will oxidize Fe² Mi back to Fe³ Mi, destroying the entire process. Fourth, cool to moderate temperatures. Too hot, the mineral structure breaks down. All these conditions once existed at Jezero Crater 3.6 billion years ago. A deep lake, neutral water, no oxygen in the sediments beneath the lake floor, moderate temperatures, iron and phosphate abundant from weathered volcanic minerals. If iron-reducing bacteria lived at Jezero back then, Vivianite is exactly what they would have left behind. And now, 3.6 billion years later, Percy might be staring directly at their traces. But there's a big problem. In 1996, NASA thought they'd found life on Mars, and they were completely wrong. How do we know this time is different? How do we avoid another false alarm that embarrasses the entire scientific community? The 6th of August 1996, President Bill Clinton stepped onto the White House balcony and announced to the world, NASA has found strong evidence that life once existed on Mars. The evidence? A meteorite named ALH 84001, found in Antarctica in 1984. Inside this 4-billion-year-old rock, scientists discovered tiny structures that look like fossilized bacteria. They were only 20 to 100 nanometers long, smaller than most Earth bacteria. But that wasn't all. They also found polycyclic aromatic hydrocarbons, PAHs, complex organic compounds often associated with life, and magnetite crystals, exactly like what some Earth bacteria create to orient themselves with magnetic fields. The entire world believed we'd found alien life. Newspapers exploded with headlines. Science fiction became reality. But 26 years later, in 2022, a new study shattered that dream. Scientists proved that the structures in ALH 84001 weren't created by bacteria. They were formed by a completely abiotic geological process called serpentinization. What is serpentinization? It's a chemical reaction that occurs when hot water meets ultramafic rock. This reaction produces serpentine, hydrogen, heat, and most importantly, organic compounds. And here's the terrifying part. Structures created by serpentinization look identical to biological structures. Same size, same shape, even similar chemical composition. So how do we tell them apart? This is where the science gets critical. To avoid another 1996 disaster, NASA developed strict criteria for biosignature evaluation, 4 key tests that must all pass before we can claim life. And the Jezero Falls sample is being put through every single one. NASA learned an expensive lesson from 1996. Now, they require 4 independent lines of evidence before claiming to have found life. First, isotopic signatures. Biological processes prefer carbon-12 over carbon-13. If the C12-C13 ratio is abnormally high, that could be a sign of life. Second, chirality, mirror symmetry. Life on Earth only uses L-amino acids, the left-handed form of the molecule. Abiotic chemical processes create 50 over 50 of both forms. If we find only L-amino acids, that's strong evidence. Third, geological context. Serpentinization leaves clear traces. Altered olivine, magnetite, distributed in specific patterns. If we don't see these signs, the abiotic hypothesis is eliminated. Fourth, multiple independent lines of evidence. Never rely on a single indicator. There must be at least 3 to 4 different types of evidence pointing to the same conclusion. And this is why Jezero Falls is completely different from ALH84001. ALH84001 was just a meteorite. No clear geological context. It could have been contaminated during its journey from Mars to Earth. We only had morphology and simple chemistry. But Jezero Falls? This is in situ observation. Percy is looking directly at the rock right on Mars. No contamination risk. We have detailed chemistry, morphology, and most importantly, complete geological context. And there's one more piece of evidence, one that few people have noticed. Remember that small olivine grain Percy found near Jezero Falls? Olivine is a mineral extremely sensitive to acid. It only exists in water with neutral to slightly alkaline pH. In acidic water, olivine dissolves within hours. The presence of olivine completely refutes the hypothesis that Vivianite was created by acidic water. This is the second independent piece of evidence showing that Jezero's environment was once neutral. Exactly the necessary condition for both life and biological Vivianite. But to be 100% certain, we need to bring this rock sample back to Earth. The Mars Sample Return Mission, PERSE, planned to bring Jezero Falls to the planet's most advanced laboratories within the next decade. There, scientists will perform analyses that Percy cannot. They'll test amino acid chirality, measure the C12-C13 ratio with extreme precision, search for cellular structures under high-power electron microscopes, and if all evidence points in the same direction, that will be level 6 on NASA's confidence of life detection scale, the highest level of certainty possible. This isn't 1996. This isn't speculation based on ambiguous shapes. This is systematic, rigorous science with multiple independent confirmations, and it all started with choosing the right place to look. Jezero wasn't selected by chance. It was chosen because every piece of evidence pointed here. So, we've journeyed from a simple question, why did NASA choose Jezero, to what might be one of the most important discoveries in human history. 60 candidate sites, years of fierce debate, billions of dollars invested, and all of it led to one ancient impact crater that was once a deep lake 3.6 billion years ago. Jezero wasn't chosen randomly. It was chosen because every piece of the puzzle fit perfectly into place. An ancient river delta, the best place for fossil preservation. Perchlorate, a potential energy source for bacteria in the subsurface safe from deadly UV radiation. Vivianite, a mineral created when bacteria breathe iron, leaving chemical traces that can survive billions of years. And olivine, proof that the water was once neutral, friendly to life. Each piece of evidence standing alone isn't enough, but when combined together, they tell a coherent, consistent story. And this isn't 1996. We're no longer relying on ambiguous shapes in a meteorite. This is rigorous, systematic science with multiple independent confirmations. But the story isn't over yet. Those leopard spots in Chiaiva Falls are still sitting on Mars, waiting to come home. The Mars sample return mission will determine their fate, and possibly the fate of one of the biggest questions humanity has ever asked. If the Vivianite in Jezero is truly a biosignature, if those spots are really traces of life, then the answer is no longer maybe. It's no, because if life emerged twice in the same solar system on two neighboring planets, then it's not a rare miracle. It's a natural law. If life exists in two places, it could exist in millions, billions of other places in the galaxy. 60 sites. One choice. One great question that humanity is about to answer. Jezero Crater might be where we finally find the proof that we are not alone, that life is not a cosmic accident confined to one pale blue dot. We are not alone. We just need to prove it.